Recording. Excellent. All right. Thank you everyone for joining today. I'm Jennifer Hammonds with the National Wildlife Federation. I am the Director of Education, and in partnership with Sharks for Kids, we have a great presentation for you today with our friend and collaborator, Sara Casareto. So sorry.
Oh, good.
If you will go ahead and let us know about the wonderful work that you do with Sharks for Kids and the personal things that you're working on to make our oceans healthier and learn more about these amazing species. Sara.
Yeah. I'm super excited to be here. I'll share my screen in just a quick second. But yeah, as Jennifer mentioned, I am here with Sharks for Kids. I'm one of Sharks for Kids ambassadors. I'm also a biologist, a marine biologist that focuses on shark behavior and shark biology. So today I'm going to talk to you guys a little bit about shark science and how we study sharks and why we study sharks. So let me see if the share screen works. It's always a fun test. Okay. Can everyone see the slides? You see it,
Yes.
Jennifer? Perfect.
Yep. All good.
Awesome. Amazing. Well, that being said, everybody, thank you so much for coming today. Today, I'm going to talk to you guys about shark science and how and why we study sharks. And there's different layers to this. But before I do that, there we go. Introducing myself a little bit more in detail. As mentioned, my name is Sara. I am a PhD candidate at Florida International University. I'm also a Sharks for Kids ambassador. Sharks for Kids is an amazing nonprofit that focuses on providing education about sharks to students just like yourselves in different formats like webinars, through resources on our website, through different events that we might be at. And so today we're going to be doing a webinar. I do all of my work out of the 10,000 islands in Florida, and I'm going to talk today a little bit just about how we as shark scientists study sharks, but also pinpoint a few, highlight a few of the methods that I use in my own shark research when I am studying sharks myself. I'm also the Margaret Davidson Fellow with NOAA, which has been awesome. It's a great way for me to get funding for my research, and I'm hopefully wrapping up my research in the next year or so, which is very exciting. So big thing about sharks that I think a lot of people know, but sometimes forget about. I know it's something, a number that escapes me sometimes, is there are over 500 different species of shark. We have a huge variety of sharks. We have from our classic predator, the white sharks, white tips, to maybe bottom dwelling sharks like nurse sharks or deep sea sharks like Greenland sharks or the pocket lantern shark. We have sharks that are ambush predators. We have sharks that are active hunters. We have sharks that spend time in the open ocean or in the deep sea or in coral reefs. There's a lot of variety when it comes to sharks, which means that the way that we study sharks has to pivot and change based off of what sharks we're studying. And also that means that not all methods are going to work in all areas. Right here, we have three very different types of sharks in different areas. And they all not only look different, but they're all different sizes. They all occupy different habitats. And this is a just a clear picture example of just even these three sharks right here. It's going to it's going to impact the way that we study sharks and how we are going to be studying them. But our biggest question when it comes to how and why we study sharks is what makes the world go round, and that is money. A lot of the time as a shark scientist, one of our biggest things that we have to think about, and it's not fun, we do it when we're in the office, when we're in the lab, we're constantly crunching numbers, is looking at our budget. One of the things that we look at is methodology, is what methods can we afford and what methods can we afford while also giving us wiggle room for error or to analyze samples or to allow for maybe trying a new method. Do we have the budget for that? So how do we get this money? We apply for grants. We work with institutions. Some people have private donors. But at the end of the day, money does make the world go round. And so a lot of shark science and the way that we choose our methods is not only based on what is the best method, but also on what is the best method given our uh, potential box that we are put into through financial costs. So the first method I want to talk about is a type of method that falls under that big shark fishing category. So I'm going to talk about a couple methods we use for shark fishing, then talk about the ways we tag sharks, and then talk about what we do when we catch sharks. So this is called a long line. A long line is used not only in shark research, but also in commercial fisheries. It is essentially one long line with lots of hooks clipped to it. Each of these hooks are baited with a type of bait. For my research, I bait it with mullet. Other people might bait it with larger bait like bonita or tuna. In commercial fisheries, this is a very common method that's used to catch for tuna. And for shark scientists, we use this to try to catch sharks. And what's great about a long line is that through one long line, we can sometimes have tons of hooks on it. The commercial fisheries sometimes have hundreds of hooks on theirs. My long lines have only 20 hooks, but that's 20 chances for us to catch a shark, essentially. So what we do is we set this long line out. We attach the hook with a tuna clip, which is that clip that you see right there, that metal clip on the actual line. It's set on either ends with an anchor, and we have buoys to help keep it afloat. What's great about these is we can set these in multiple different types of habitats, on seagrass beds, on coastal habitats, and open oceans, which can give us a large diversity in what types of sharks we might be catching. The other type of method that we use is a drum line. And so we use what's called a bottom set drum line, which is there's a weight that sits the bottom. Attached to this, there is then the actual fishing line or a monofilament, in which there is one singular hook, which has a bait attached to it. Then again, attached to the weight, we have a line that goes all the way to the surface in which there is a buoy. That's so we can recover and actually find our gear again. What's really great about drum lines is that because the fact there's only one hook on a drum line and the way we attach it using a swivel is that it actually allows the shark to keep swimming in circles, which is a really great method for us to reduce stress on the shark. As some of you guys might know, sharks need to just keep swimming. A lot of shark species need to swim in order to continue to get new oxygen and in order to breathe. So drum lines, although we lose the extra chances from a long line, like with a long line, you can get 20 plus or even more chances. For a shark with the drum line, we set multiple, but we might only set 10 at a time. So only about 10 chances for catching a shark. It's great though, because it's a lot lower stress and it's really useful when we're catching those larger sharks. So what we do is we put the bait out into the water. We have that weight attached to it, so it's at the bottom, and we have that buoy so we can recover it. Then we check our lines. We'll check our long lines. We check our drum lines. We bring it all back in the boat. And if we don't have a shark, we will just redeploy that gear and wait a little bit more soak time, anywhere from an hour to 2 hours, depending on where we're fishing or what type of shark we're trying to catch. But if we have a shark, that's when we go into what's called our shark workup. And so right here, you see a perfect example of one of the first things we do in our shark workup, which is measure our shark. We've caught our shark. We've pretty quickly figured out what kind of shark this is. Right here, we have a beautiful lemon shark, and then we will measure our shark. When we measure our shark, we take 3 measurements. We take our pre-caudal length. That's our first measurement. That's from the tip of the nose all the way to just before the caudal fin or the tail of the shark. We then take our fork length, which again, tip of the nose, all the way to just where that tail goes into the fork, so right here. And then we have our total length, which is the measurement from the tip all the way to the end of the tail. Some people also do a stretch total, which is where you keep it flush along the body and you stretch it all the way to the tip of the tail. The reason we do these 3 different measurements is because sometimes we have sharks that have damage on their tail, so you don't actually have the full tip of their tail. So having the fork length or their pre-caudal length is a great alternative measurement. And 2, not all shark scientists and institutions necessarily will measure the same way, but will usually get at least one measurement in common, which allows the opportunity for us to compare our data with data sets from years in the past, or even data sets from other countries and other scientists. And we can still find some common measurement usually at that point. So we take all 3 measurements when we catch the shark. And then at this point, this can help us determine maybe how old the shark is. If we catch the shark again, we can see how much it's grown, or if someone else catches a shark, we can see if it's grown since then. So here's an example of us taking that pre-caudal length of a bonnethead shark, tip of the nose, all the way extended to just before the tail. Then we will often determine if the shark is a male or a female. And the way we do that is we actually turn the shark over and we look between their pelvic fins. And if it's a female, they don't have any extra appendages in between those pelvic fins. They just have the opening for the cloaca as shown here on the leftmost image. But if it is a male shark, they have what are called claspers. And claspers are two sexual appendages found between those pelvic fins. And they're very obvious and apparent. And it's nice because we can easily see externally if a shark is a male or a female. This is important because we've figured out that some sharks will actually separate themselves within their population into male and female populations. They'll spend times in different areas. And also with male sharks, using claspers, we can determine if they're sexually mature or if they're not sexually mature. We will often also collect samples from the sharks. One of the most common samples that we collect is a fin clip. And so with the fin clip, we can do a lot of different things. It is a small clip of the fin, equivalent to taking a nail trimming or a hair clipping from their dorsal fin, which is that top fin, as you can see in the top left, you have someone collecting it. We can put that in ethanol and use that for actual genetic analysis. This can help us figure out how related different shark populations are. This can go into genetic databases that can be used for future conservation work, like determining whether a shark fin on the black fin on the black market belongs to a certain species or protected species, or just going into fisheries or grocery stores and determining are things actually labeled appropriately or not, and where is that miscommunication happening. But we can also use those fins to the fin clippings to determine their diet and where sharks are feeding, what they're feeding on. This also goes into different samples that we can collect. We can collect muscle biopsies, which is just like if you go to the doctor and have to get a biopsy done, we can take a small muscle plug. This can be used to measure things like mercury accumulation, accumulation of pharmaceuticals, like uh, we've seen recently in the news, you might have seen that there's been antidepressants found in fish or other PFAS or buildup of plastics and other sorts of heavy metals and toxins. And we can use muscle to figure out, is that prevalent in these shark populations that we are sampling? And then we can also collect blood samples. And blood samples can give us a lot. We can use these to determine diet. Using stable isotope analysis, we can figure out, generally speaking, where in the food web they're feeding or in what food web they are feeding. Are they feeding in a coastal food web or in a estuarine food web or in a food web that's based in seagrass beds? We can also use blood samples to determine things like uh, whether or not they have certain immunoresponses to different toxins. And that's a field that's still being studied, but it's pretty cool. And just like when we go to the doctor and they get blood and the doctor can figure out a lot of things, we can use the blood samples that we collect from sharks to figure out a lot about the health or the stress of the shark as well. Another type of sample that we collect are fecal swabs. This is also known as a cloacal swab, which is, as I mentioned earlier, with the cloaca, it's between the pelvic fins. We can insert a Q-tip, similar to like what they did with COVID tests. We put it in the cloaca swab, swab it around a little bit, and actually send that into the lab. And using this, we can use genetics and the DNA that we got on that fecal swab to determine what the sharks are feeding on. And this is great because historically speaking, and this is still a method that's used, but the traditional methods for determining diet for sharks involves either euthanizing the shark and actually doing a dissection of their stomach, or doing what's called a gastric or stomach lavage, in which we use water to gently flush the contents of their stomach back out, and then we have to go back in the lab and sort through it. This is a little less time consuming, more expensive, but a little less time consuming and can cause less stress on the animal, which is really cool. This is also a method that we're still developing and still fine tuning, but pairing it with methods like the stable isotopes that I mentioned with the blood or the fin, we can get a nice clear picture of what the sharks are feeding, which is pretty awesome. And then we go into a methodology that I think everyone is pretty familiar with if you've watched anything on Blue Planet or National Geographic or Discovery Channel, which is tagging sharks. And this is where a lot of times that money and cost comes in, because the type of tag we use depends on the type of question we're asking, but also depends on the cost. So this right here is a pit tag. This is very similar to the microchip you might put in your dog or your cat. It's a small pit tag, as you can see in the bottom right corner, sit in the hand, and it goes just under the skin. And we will then use a reader to see, and it'll come with a unique serial number right there. This is great because it just kind of lives there. It doesn't bother anything. And we are able to figure out, if you read it, if it pings, if someone else has pit tagged it, or if we've pit tagged it before. One downside is that there's no external thing, which is why a lot of the times when we pit tag a shark, we'll often still put an external tag so that way we can visually see if the shark has been tagged before as well. So here's an example. We are checking it. We just pit tagged this shark in part of our project in this is probably either the Bahamas or Turks and Caicos, in which we pit tagged a juvenile lemon shark and we are getting ready to release it. And we're just checking to make sure that tag is actually coming up on our tag reader. Then we have another type of tag. These are those external tags I was talking about. So right here we have a roto tag that is visible. The rototag is just like putting an earring through that shark's dorsal fin, and it has a unique number on it with usually contact information on the other side. This is great because if anyone sees or catches this shark, they can reach out to us and say, hey, we caught this shark. It had a tag on it. The tag number was B6709. This is where we caught it. We kept the shark. We're actually going to eat it. Okay, great. We know it's no longer in the ecosystem. Or they'll be like, hey, we caught the shark. We actually measured it and released it. Great. Now we know that, oh, it's grown since we caught it a few years ago, or, oh, wow, it's been staying in the same area, or it moved and it's in a completely different area. Another type of external tag that we can use is a spaghetti tag that gets inserted just at the base of that dorsal fin, right on the top of their back. I use both rototags and spaghetti tags for my work. And I mentioned earlier about how we can catch a shark and figure out they've moved. We caught a tiger shark a little bit ago that I'd caught a tiger shark just off Palm Beach that already had a spaghetti tag in it. And when I reached out, found out that the shark was previously tagged 3 years ago and it was tagged in Georgia. So it had swam a little bit south since then. And who knows what it's done in the time frame, but that meant we know it's at least coming to Florida. And it had grown since then, which is pretty cool as well. Then we have some of our more expensive tags. These are our acoustic tags. These are great for when we want to start tracking our sharks and seeing a little bit more fine tune into their actual movement and where they're spending their time. So these are examples of the acoustic tag. What we do is we create a small incision on the stomach of the shark on their ventral side. And what we do is we insert that tag into their body cavity and then we stitch them right back up. This time, anytime the shark with that tag swims by, it'll ping and we are able to see where it is. This kind of tracking method of a receiver and pinging is pretty used. So here is an example of what is a receiver. So I showed you the tag. This is the receiver. So with acoustic tags, it's a two gear method. You need something to listen for that acoustic tag, but you need the actual acoustic tag to be in the animal. And the type of tag you have, the size varies on the size of the animal. You can have tags that can last upwards of 9 years. We have tags that maybe only battery lasts about a few years, maybe 5. But anytime that shark swims near a receiver, as shown right below, that receiver will take that ping, and then we actually will bring it back up. As you can see on the top left, it's a little bit dirty, but that's us bringing that receiver on to download that data. And we'll be able to know, oh, shark A swam by this receiver on this date at this time. And you can use this to figure out their movement. You can figure out social behaviors in sharks. Shark A and Shark B are always together, or Shark A and Shark B always go away if Shark C shows up. What sort of social patterns are we seeing there? Which is pretty cool. So here's an example of like a graph, as you can see. The acoustic receiver is listening for that ping that acoustic transmitter is letting off from inside of the shark, which is pretty cool. And here we have a nice video that really, really shows it. It's recording that data and it keeps recording it. So anytime a shark swims by, a new shark, a different shark, or the same shark, it'll re-record that. We can figure out if a shark is staying in an area for a certain time frame, if they're hanging out near a receiver, or if they swim away. Then we have what are called our pop-off satellite tags. So these tags, we put them on the dorsal fin, and what they do is they are attached to the dorsal fin, and they are tracking the movement of the shark the entire time that it's on the shark. And so it can track things like how deep the shark is, where it's swimming, latitude and longitude. Some of the more fancier ones will have attachments to them, like salinity measurements or speed measurements. They get added to that pop-off satellite tag. But the shark will be swimming with this tag attached to it. and it's recording data that whole time. It's recording where they're swimming, how deep they're swimming, what time it is when that is happening. And then after a certain amount of time, that tag will actually pop off, come back to the surface, and it will transmit a signal. And we will go out. And once we get that satellite signal, we will go and retrieve that tag, download all of that data. Then we have our satellite tags. So these tags do not pop off. We attach them to the dorsal fin, and they can last a really long time, which is great. And they can give us some really cool data. These are great for sharks that are more coastal sharks or sharks that will come to the surface. So this is not a type of gear we would use on a deep sea shark, but these are great to use on white sharks, tiger sharks, those sharks that we know come to the surface of the water at some point and pretty often compared to other shark species. And this is great because we can use this to track the location of a shark as it swims, especially as it swims long distances. So here we have the track of a shark. Every time that shark came near the surface of the water, it was able to transmit that satellite signal. So if any of you guys have ever looked at shark tracking online, a lot of the times it's these satellite tags. And you can see that we can really get a good idea of long distances. And this doesn't require us to go out and try and find it, because this is great for sharks that have huge migratory patterns that maybe are going to travel really far distances in a long time frame. And we want to be able to track that. And we can see every time that shark pops up, we get a little bit more data about where they're spending their time. We can see areas in which they're concentrating their time frame, where they're spending their time for long periods of time or coming near the surface a lot, which is pretty cool. And every time it comes and breaks the surface, we get that data from the satellite. So here again is that visual of it coming to the surface, it transmits, and then we get that information because it goes to the satellite, and then the satellite will send us that information back. Yeah, perfect. Then we have our non-shark tagging type of method that we at Sharks for Kids have used and other scientists use as well. This is called BRUVing, or baited remote underwater video systems. It's technically a noun. We definitely use it as a verb, like I just did with BRUVing. What is great about BRUVs is that you can build a BRUV out of PVC pipes and some dive weights, or you can build a BRUV that is foldable out of stainless steel. You can build BRUVs that aren't foldable and are just affixed and are really nice and heavy. You can build floating BRUVs that are great for pelagic work. And what's wonderful about these is that they can be deployed across any type of habitat. They're very cost efficient, and they are non-invasive very, very non-invasive. All it is it's a frame that has a camera attached to it, a GoPro. So you can use a GoPro too, and it still gives you the same amount of information. It has a bait arm that extends from that frame. And at the end of that, there is a bait cage, which is filled with some yummy, delicious, oily, bloody fish. And for the time frame of an hour, 2 hours, however long you need it to be for your research, it just sits there and it records. And at any time anything comes up to it, we'll be able to see it on the camera, which is really great, especially because we can use this sort of method across different areas. This can be used by scientists all over the world. This can be very comparable, which is great. We can compare this with different BRUVs done in all over. Because as long as you have the camera, a bait cage, and bait in there, you can film it and you get it. You can get data and compare it all over. So what you do is you set it down. As you can see, this is a shallow water habitat with a very tidal system. It does have a line with a float just to make it easier for us to find again. And we will just let it sit there, and anything that comes up, that camera will catch. What's great about this is we can also see behavior a lot of times from this. So for example, here we have a little lemon shark that is checking out that bait cage that you just saw get deployed. And here we can see behavior of the shark actually coming up to the cage. But you can also see the behavior maybe of other fish and see how we have that little lemon shark that actually gave way to a bigger lemon shark. We can kind of see a little bit more of that deference behavior. Here's another example. We have some fish hanging out by the bait cage. We're in a seagrass bed here. And we would sit here and just watch. And as we come in, this nurse shark comes in. The fish are going to move a little bit out of the way. We got that nurse shark that's coming and checking out the bait cage. Definitely trying to get some of the yummy goodies on the inside. And when we're watching this footage, we will watch it and we will look to see what species of sharks come up, but also how many. This can be great for estimating abundance. And it's not just in seagrass beds or in shallow waters. We can deploy these in areas like coral reefs. As you guys might have noticed, we had that beautiful great hammerhead come in. Notice on the second dorsal fin, it's actually missing its second dorsal fin. That would be something we would note. We would note unique injuries or morphologies like that, because then if, hey, we see that hammerhead again later, it's pretty unique. We might be able to be like, huh, there's a really good chance that it might be the same shark. Let's keep an eye out for that. It's pretty cool. So why do we study sharks? I talked about how we study sharks. Why do we study sharks? Outside of the obvious fact that sharks are pretty cool, sharks are unfortunately facing a lot of stressors. About 100 million sharks each year are killed by humans. That's a very large number. Some of this is intentional fisheries, but some of this is through bycatch, which is unintentional catching. There's other stressors for this. We have overfishing, which is a big, biggest driver with 1/3. Some of it is intentional, some of it is not. We have climate change that can be that is a cause for why sharks numbers are decreasing, loss of habitat, pollution. And so studying these sharks is super important because not only does it help us understand sharks as a whole, but it helps us understand how these different stressors might be impacting sharks in different areas. If we know how sharks are doing in one area, we can then determine, oh, sharks are doing better somewhere else or they're not doing better here. What is the difference? For example, there's been a huge global project using BRUVs, those beta remote underwater video systems, where they've actually been able to pinpoint areas in which sharks are doing better compared to others and figured out like, oh, wait, those areas have marine protected areas. They're protecting the habitats of the sharks, which is why sharks seem to be doing better here compared to other areas. Sharks are declining and they are threatened. A lot of shark species are threatened. We have a lot of species that are endangered or critically endangered. So getting a strong and firm understanding of the different impacts that are on sharks and understanding sharks is crucial so we can ensure that oceans stay balanced. I mentioned bycatch earlier. Some of it is intentional, but unfortunately we do have situations like bycatch where you have maybe people going out and they're trying to catch tuna, but unintentionally they're also catching things like turtles or dolphins or sharks and rays. Climate change, some of the things that we need to study and also understand the impacts of are things like water temperature, storm frequency, coastal erosion, sea level rise, the ocean chemistry, whether it's acidic, weather patterns. We've found that weather patterns do change with climate change. We know that sharks in certain estuaries will actually leave if a storm is coming. They'll go to other systems where the impact may not be as strong. And so knowing that is important because we might be able to be like, whoa, the sharks all left, but maybe it's not that they're declining. Maybe they've actually moved systems. Or, hey, we know they actually didn't leave the system because we don't have any acoustic tags and they're not pinging anywhere else. And knowing that, okay, we had an impact here and we need to see what happens maybe next year with the new storm season. And then we have our perfect, I think this is a good visual of pollution. I think sometimes we forget that our planet is 70% water. Trash, sometimes it seems to always find a way back into the ocean. And a lot of the times that might not seem as big of a deal as we think it is, but it is a big deal. We have plastic, kind of seems to last forever. Fishing lines can get wrapped around animals and that visual as you see. And it can cause a lot of damage, especially if they don't break. A lot of animals will ingest plastic and then it'll keep their stomach full and then they actually starve with a full stomach, which is tragic. That to be said, that is why we study sharks. We have this pollution and that's something we can do in our day to day is reduce how much plastic we use, reduce how much trash we create. And if we are out on the beach, I always like to encourage my friends and family and my interns and volunteers that if you go out and you see trash, take at least one piece. Take your trash with you, but then take an extra piece. If everyone just takes an extra piece of trash home with them every time they're out, the world would be a much better place. So sharks need your help. As I mentioned, there's some really easy, tangible ways we can help sharks through reducing our pollution, reducing how much trash we create, uh, telling our legislators and telling our policymakers that we care about sharks and we want policies in place that protect sharks and protect their habitats is super important. And then with Sharks for Kids, we are spreading education. We work closely with different organizations all around the globe, and we do work closely with sharing our research and sharing shark knowledge with people all around. Connect with us. Go on our website, email us if you want us to come into the classroom, email us at info@sharksforkids.com. You go on our Facebook, our Instagram, our Twitter. We have TikTok. Um, any of these ways are a great way to reach out to us, learn about sharks, have other people learn about sharks. I want everyone so obsessed with sharks and obsessed with Sharks for Kids that it'll be a thing that you say Sharks for Kids in a room and everyone should know what it is, you know? That being said, thank you all so much. I really appreciate it. And Jennifer, I think if there's any questions, I am happy to take them.
Excellent. Thank you. I've been trying to put some resources in for our participants. And so be sure to check those out. Let me move some things out of the way. I can see. All right. As you're coming up with questions from students, feel free to put them into the Q&A. We do have a couple. So have you ever used BRUVs in your current research?
Yes, so that's actually a great question because it's twofold. I've been involved in BRUV projects that use BRUVs successfully and have been great. For my PhD work, I actually originally wanted to use BRUVs to look at the more smaller shark movement, like the juvenile shark movement. And so I did deploy a few BRUVs, and then I realized that in my system, the water is chocolate milk equivalent. And so it actually doesn't work. It's just too murky, and so it wasn't an option. So I actually switched to long lining for that reason. But I did go with BRUVs first because they are a little bit more cost efficient, and I can get some more unique things like getting behavior out of it, which would have been pretty cool. But that's part of pivoting as a shark scientist. That method did not work in my system, so I switched to long lines instead.
Nice. Great. All right. So speaking of your research, what is your research question?
Yeah. So my PhD overarching big question is I am studying and looking at the factors that influence the decision making and habitat use of the juvenile sharks in the 10,000 islands of Southwest Florida. So the area where I'm looking is a multi-species shark nursery. So there are a lot of different sharks that are likely using this estuary as a nursery habitat or an area for the sharks to spend their time while they're juveniles and growing. And I am interested in not only what factors make this a nursery, like is it salinity, is it temperature, what is driving what's spending their time, but I also wanted to tease out some of those more biotic factors. So one of my questions is looking at how predation risk or the chance of encountering a bigger shark or a predator might be a driver for nursery use. And for that, that's why I'm using those drum lines is they selectively target those larger sharks and seeing how risk might change across the system and how that might pair with also our catch rates of those smaller sharks. I also, with it being a multi-species nursery, I also am interested in how the foraging habits might vary within or between different species in the system, which is why I collect those blood samples, the fin samples, and those fecal swabs to figure out. It's hard to figure out competition, so I don't want to say competition, but at least determine where and what these different sharks are feeding on and in, and to see does that change? Does it change seasonally? Does that change as the shark gets bigger? Does it change within a species? Does it change across species? Does it change based off of where I've caught the shark? If I caught the shark in our back bay, so further into the mangroves compared to on the coastline, is that gonna, are we gonna see differences in those results or not?
Nice. Great. Sticking with your research, we have a tag question. Do you use specific tags currently? And if you do you have a preference?
Yeah, so I currently use, I'll actually pull it back up, the visual, just so you guys have it. I currently use the spaghetti tags and roto tags for my research, largely because I spend a lot of time out there and I've caught and tagged over 300 sharks over the course of my past 2 years. So I use these tags, the roto tags, on my really small sharks, and then I use these spaghetti tags on my larger sharks. I prefer the spaghetti tags between the two, just because I find that they're a little more reliable. They don't necessarily pop off as much. It's hard for them to do, but as the shark gets bigger, a lot of the time that fin will get a little bit thicker and it'll just pop off, which is fine. That happens.
So
I
it
find the
loses an
spaghetti,
earring sometimes,
yeah, it loses an
like
earring,
we do.
The spaghetti tag I find is a little bit more reliable. And it's been pretty cool. As you can see, even on the bull shark down there, algae will grow on it. So sometimes if we catch a shark, we have to wipe that algae off, but we can see that number again. Uh, I have caught sharks again, the same shark actually in my system that I tagged a few months prior. And I was like, you didn't learn your lesson. You're still here. You're still biting my hooks. So, okay, I'll collect more samples, which is great because then I can compare my data. But future work that I'm potentially interested in for even the same system, it would be maybe adding some acoustic tags. Based off even just my spaghetti tags and my rota tags, we are catching the same individuals. Uh, and I've caught sharks that other researchers have tagged there years ago. So it'd be interesting to see if maybe if we added some acoustic tags and receivers in the system, how long these sharks might be actually staying, and do they move within the system? Do they leave and then come back? Where exactly are they spending their time, which would be really cool to see at a fine scale. But for my dissertation and for, as I mentioned, cost, spaghetti tags and rota tags were the most efficient thing. I get my spaghetti tags through NOAA, so they're actually free for me, which is great. And then my rota tags are from the lab, and they only cost like 25 cents to make each, which is really nice. Versus some of those fancier tags can cost anywhere from a couple hundred to $20,000, depending on
Oh,
what
wow.
is attached to it. Yeah.
Wow. That's amazing. So we're going to stay with your research. It sounds like a lot, you've seen, encountered lots of different species of sharks. Would you say you have like a top three you see most, or it's
Yes.
really very varied?
So definitely, we definitely see more of the sharks that are using it as a nursery habitat. But because I've started drumlining this system, we've determined that there are definitely larger individuals that will also be there. So I would say, based off of my fishing, the top three sharks that I've seen in the Rookery Bay, 10,000 Islands region is lemon sharks, bull sharks, and blacktip sharks. That being said, I do know that there are a lot of bonnethead sharks in that system, which is the smallest species of hammerhead. I have caught zero with my research, but another fishery scientist that I partner with very closely, he uses a completely different method than I do. It's called a gill net. Um, and he catches a lot of bonnetheads with those. That's why also as a scientist, the type of method you use matters. Because if I didn't know, or if this scientist wasn't fishing the system and I didn't visually see them and I didn't have his data to compare to, I might not even know that there's a lot of bonded heads in the system. But because I partner with the scientist, he uses a different method, we've been able to figure out like, oh, my methodology just does not target bonded heads very well versus the gillnet does. And so that's why it's really important that you keep your blinders open and you keep like the idea of methods really matter.
Nice. So you made me think, hearing you speak about methods, and putting my teacher hat back on. When you think back to high school, and you think about the scientific method and the way that you did science in high school or maybe even early college, what would you say might be some of the most useful skills that help to lay some of the foundation for where you are currently?
I think it cannot be overstated the importance of appropriately identifying your variables. Even now as in a PhD, one of the questions that like my committee and my advisor will always go is like, what are you asking? What is your hypothesis? And is the methods and is this experiment you set up actually testing that question? And that's something I always give to people as an advice. If you know your variables, one, if you can identify, okay, I want to test salinity, I want to see how salinity affects where the sharks are swimming, well, then you need to find a way to measure salinity. And then you need to find a way to figure out where the sharks are spending their time in relation to that salinity. You can do it local, like, okay, I'm measuring the salinity where I'm fishing. If I caught the shark here, that means they're present in the salinity levels. Or you can have those really fancy tags, which measure salinity at the same time, and it pops off after a few days, and you can track if salinity changes as they're swimming and how much time they spent. There's a lots of ways you can test that hypothesis. But if I go out and I just catch and tag sharks and I never measure salinity, then I'm not answering that question. And so that's really important is you really need to narrow down the variables you want to test and your hypothesis. And another thing, and this is something that's important even now as I'm getting into more of my writing phase of my dissertation, is read scientific papers. I know it's something like everyone is like, oh man, but I want to go do the science. You don't know the science that has been done before you, and you don't have a strong grasp of the theory, you might not really understand what you're looking at. And so it's really important to read the science that people have done before you. One, because it's cool science and they put it out there. And two, it can inform your decisions and the methods you want to use. And it can also inform how you're going to approach a problem.
That's great advice. Thank you so much.
Yeah.
Let me see. Oh, challenges. What
Ah,
challenges have you met? I'm sure there's always challenges, but maybe most recently, what's been a recent challenge you've had to overcome?
yeah. I'll say a big challenge just in general is finding funding. You can have the coolest project in the world, but if you don't have the funding for it, you can't do it. Um, and so that can be something that I think is like scary when you're going into the field. But there are lots of amazing grants out there. There's a lot of amazing programs. You can work closely with advisors or with other scientists to apply for grants and get those funding. I always recommend go outside the box. Some of my funding has come from my college sorority or from, I'm an Italian American. I got funding from the National Italian American Foundation. I got funding from the Marco Island Shell Club because they just wanted to support students doing research. There are ways outside of maybe traditional methods that you can always go. And so always think outside the box. I'd say recent challenges is I guess one of the things is just being organized. Sometimes when you're juggling so many moving parts, it's easy to drop a ball. Uh in the 2 years that I've been more than 2 years that I've been fishing in the system, I have not once forgotten to check if I had the boat available. And I forgot last week. And you know what? It happens. And it's a small, silly thing because at the end of the day, it's not a big deal. I'll just schedule for another day. Uh, but if you don't go into it with like the mindset of being organized and being communicative, um, it could be if it's a reoccurring issue, it could be a real detriment to your project. Because I've never had a miscommunication issue like that in the past. The scientist whose boat I was using had no issue to be like, don't worry, we'll just go next week. I don't mind in like pivoting last minute because it's the first time it's happened. It occurs. And I think I hit on another point, communication. It is always better to be over communicative than under communicative. One of the things I do, even with interns or volunteers in the field, is I always assume that people don't know anything. And I always disclaim it. It's like, hey, my modus operandum is I assume you don't know anything about what we're doing. So I'm going to over explain. It's not me trying to talk down to you or assume you don't know anything. It's me just wanting to ensure we're all on the same page because I don't ever want to be in a situation where I think they know something and they don't. And then something dangerous happens or someone gets hurt or the sample that we need to collect doesn't happen or the data doesn't get written down. I would rather just everyone be on the same page from the get-go.
Mhm. That's great. And I hear throughout your talk, I hear the science, the technology, I hear the engineering components, I hear the math components, but what I also hear are very important are your reading skills, your writing skills, and your
Mhm.
ability to communicate with others are just as important as those hard STEM skills that we talk so much about and
Absolutely.
teach so much about in school. So, well,
Yeah, I
that
think
is all the
a well-rounded
questions, but
scientist is
yes, go
the best
ahead.
type of scientist. And it's interestingly enough, but like you could be great in the field and great with mentoring and collecting the data. And if you have an awesome friend who's really good at statistics, you don't necessarily have to be the best. You can reach out to that amazing friend and be like, hey, you want to help me with my statistical analysis? You'll be a co-author on the paper. It's about working together and collaboration a
Yes.
lot of times.
Yes, that collaboration too. So important, so key. I know a lot of times in school we focus on individual contributions to outcomes, but being able to collaborate and build together something that relies on the strengths of each individual to deliver something amazing
Yeah,
is also highly
absolutely.
valued.
Shark science is team science. You can't go out, catch a shark, tag a shark, do a workup if it's just one person on the boat. It's just
Ah,
literally not safe. But when you have an
fantastic.
amazing team of people, you can do it.
Yes, yes, for sure. All right. Well, thank you so much, everybody, for joining today. Don't forget to grab the resources from below before you head out. Again, we hope that you enjoyed this opportunity to hear about shark science with Sharks for Kids. Thank you, Sarah. We hope that you consider, if you're not already, joining Eco Schools with National Wildlife Federation. And everyone have a wonderful afternoon.
Thanks, everyone.
Bye.